Hello everybody, this is going to be my second video of the series in acute inflammation, everything you need to know. So in the previous video we were discussing about the receptors and I told you the role of antibodies, right? I told you how antibodies work, this point. So our phagocytes also have receptors for Fc portion of antibodies and for complement proteins. Let me explain you this complement protein thing. Remember I told you about opsonin. Opsonin are the molecules which make the phagocytosis of certain substances easier. How do they make easier the phagocytosis easier? Remember one thing. You have a well-known opsonin, complement 3b, in your body. Okay, complement 3b. Suppose this is an antigen that has entered the human body and you have C3b in your body. Okay, C3b binds this antigen and your phagocytes have special receptor to recognize this C3b. Okay, now suppose that our phagocytes recognize this thing. This C3b binds the receptor on the surface of phagocytes and the phagocyte will engulf all of this thing. It will engulf all of this thing and it will eventually destroy. So my question is, how opsonin make phagocytosis easier? It's because they bind certain type of offending agent very tightly and then phagocytes easily capture it and break it down. Is this clear now? Okay, so I think this is also clear. So we have two types of opsonins, mainly two types of opsonins in our body: antibodies and complement proteins. Okay, then you have some other kind of receptors known as monos binding lectin, which recognize microbial sugars. Okay, once they recognize microbial sugars, they will activate your complement. That's why they are known as collectins, because they are activating your complement system. Is it clear? So what kind of receptors do we have in a body to recognize offending agent or a foreign substance? If you have the receptor on the plasma membrane, it's going to recognize extracellular antigen, and within the cell in the endosome, the receptor will recognize intercellular antigens. Then you have nod-like receptors, obviously toll-like receptors for microbes, particularly bacteria. Then you have nod-like receptors, rig-like receptors. Then you have certain receptors which recognize indicators of cell damage. Then you have receptors for Fc portion of antibodies and for complement proteins. Then you have monos binding lectin, okay, which are known as collectins. I hope this is clear. Look at this image and try to recall the definition of opsonin. What is opsonin? Opsonin is any molecule which makes the phagocytosis of certain substances easier. And just remember the this role of C3b. Okay, what happens once our body has recognized the antigen? What happens then? How the cell that has recognized an antigen or offending agent is going to elicit the inflammatory response? How? This is very important. Okay, suppose this is your liver and liver has certain kind of offending agent, maybe a virus like hepatitis A, B, C, or it has suffered some kind of injury, hemochromatosis from iron, or if the person has Wilson disease, copper has created some kind of injury, whatsoever is the reason. Suppose the hepatocytes or Kupffer cells recognize hepatocytes or Kupffer cells or whatever kind of cells present in your liver recognizes this offending agent, okay, by certain receptors which we have already discussed. These hepatocytes and Kupffer cells will start secreting some kind of special substances. Okay, some kind of special substances. I will tell what kind of special substances they will secrete. Okay, these substances will act upon the vessel wall, and as well as they will act upon the leukocytes. Clear? So our next step is to learn about these substances. What are these substances? These substances are cytokines and chemokines. Cytokines can be interleukins, tumor necrosis factor alpha, interleukin, particularly interleukin 1 and interleukin 6. These two are very important, okay? Then it can be tumor necrosis factor alpha, it can be histamine, it can be bradykinin. Remember, bradykinin has major role in causing pain. So another cardinal. Then you have leukotrienes, okay? Then chemokines. What are chemokines? If I simply ask you, what are chemokines? Chemokines are substances, are the chemicals which attract phagocytes, which guide phagocytes. Where do they actually have to come? This is liver, and liver is secreting a lot of chemokines and blood vessels. When the phagocyte finally comes out of the blood vessel, these chemokines will guide it through that where do they actually have to come. Chemokines are the attractant, are the phagocyte attractant. Okay. Then chemokines can be of two types. They can be exogenous, they can be endogenous. Exogenous, like this is very funny thing. Like bacterial peptidoglycans and lipids are attracting chemokines. Can you just imagine the bacteria is itself calling its enemies? Like, okay, so exogenous chemokines are bacterial peptidoglycans and lipids, and endogenous are interleukin 8, C5a. Remember I told you a moment before, C3b is an opsonin, whereas C5a is a chemokine. Okay, then you have arachidonic acid metabolites like leukotriene before. So these are the substances. These substances are cytokines and chemokines. Is this clear? Okay, so this was the another step of inflammation. Now we will come to next step. We will learn how do cytokines bring changes in the vessel wall. If they actually want to bring leukocytes out of the vessel wall, we need to bring some kind of changes. That I told you already, but I'm going to talk about that in detail now. Okay, suppose your liver secreted cytokines and chemokines. Okay, majorly cytokines have a role in this thing. This was your vessel first. Now these substances will first bring vasodilation. Okay, is this clear? First thing will be vasodilation. We want to dilate the vessel. Why do we want to dilate the vessel? Why? Because we want more and more blood to come to the liver so that we can have more and more leukocytes towards liver. This is clear? And one more thing, by creating vasodilation, we will able to create stasis. Okay, the blood will be flowing at a very slow rate. What is the benefit of that? Its benefit is we will have plenty of time to capture these leukocytes. We want to bring them to margin. Is this clear? We want to bring them to margin, and this way they will be able to come out and go to the liver. Is this clear? So first thing is vasodilation, and I told you how vasodilation occurs because of cytokines. What kind of cytokines particularly? In causing vasodilation, you have three major cytokines: histamine, bradykinin. Remember I told you bradykinin also has role in pain, but it also has some role in creating vasodilation. Then you have leukotriene. This is clear. These three cytokines will create vasodilation. Clear? Okay, what happens with vasodilation? Okay, we want vasodilation because we want more and more blood to flow towards the liver. We want more and more leukocytes to come to in the liver. But what will be the signs of vasodilation? Very important. With vasodilation, you have more and more blood, you will have redness. The tissue will become red in which inflammation is going on. Okay, it is known as rubor. This is one of the 4 cardinal signs of inflammation, rubor. And with more blood flow, you will have more heat flow. So the tissue which has inflammation going on will be warm. And this warmth is known as calor. This is another cardinal sign. Okay, and pain, which I already told you, pain because of bradykinin will be dolor. Or, okay, dolor. Okay, next thing. So we discussed vasodilation. Then these cytokines increased vascular permeability. They want to increase vascular permeability. How do they increase vascular permeability? There are 2 ways of increasing vascular permeability. Suppose vasodilation has already occurred. The most important way of creating the increased vascular permeability is particularly histamine. Particularly histamine comes and increases the contraction of these endothelial cells. These endothelial cells contract. When they contract, they leave spaces between them. Okay, they leave spaces between them. And when spaces are there, fluid will escape out from these spaces first. Fluid, protein, and obviously leukocytes, they all try to escape out. And this process is known as exudation. Okay, exudation is escape of fluid, proteins, cells from the vessel into the extracellular compartment. Exudation. So what will be the sign of exudation? Very, very easy. Whenever there is increased fluid in the extracellular compartment, we call it as edema. So when there is inflammation, there is edema because of increased vascular permeability. And this sign, this cardinal, this is the 4th cardinal sign. This is known as tumor, swelling. The tissue in which inflammation is going on will have 4 cardinal signs: redness, rubor, warmth, calor. These 2 are because of vasodilation. Pain is due to bradykinin, dolor. Then edema is due to increased vascular permeability. This is tumor. Is this clear now? Okay. So I was talking about these cytokines. Okay. These cytokines created vasodilation, and now they have created vascular permeability. They have increased the vascular permeability. Let's read the text. Okay. We have already learned vasodilation. Vasodilation is because of histamine, bradykinin, and leukotrienes. With vasodilation, you will have stasis. Okay, and then there will be increased vascular permeability. How? There's retraction of endothelial cells, and then there is endothelial injury. Okay, let me explain this to you. In later stages, when leukocytes bind here with certain substances, I will teach you what kind of substances I'm talking about. Just listen to me now. When leukocytes bind to these endothelial cells, at first they have very weak connection. With blood flow, they detach, they attach, they detach, they attach. Okay, they detach, they reattach, they detach, they reattach. This attaching, this detaching, and this reattaching, this process is known as rolling of leukocytes. Okay, rolling causes some kind of injury to endothelial cells. When endothelial cells are injured, obviously you will have space. If I just injure this endothelial cell, obviously I have space now. I have increased vascular permeability. This is another concept, just you should know it. Major thing is this thing, retraction of endothelial cells. Then there is exudation. I told you that as well. Okay, then very important concept is coming. This is very, very important. Very, very, very important. Okay, let me just clear all of this so that I can explain that to you. Do you remember I told you that we have to bring leukocytes out of the vessel wall, and we first bring them at the margin by escaping the fluid out of the vascular compartment, so that phagocytes are somewhat at margins. Then first thing is obviously the escape of fluid, that is exudation. Then we require some other things as well to bring leukocytes out of the vessel wall, which are, okay, listen to me carefully, okay? This is your vessel wall. And on the vessel wall, you have endothelial cells. And they have spaces because you have increased vascular permeability by retracting the endothelial cells. Now, fluid has also escaped out of the vascular compartment, and leukocytes are somewhat at margin, somewhat, okay? Now you want to bring them here. You want them to attach over here on the surface of this vessel wall, so that they can easily escape from here. So what thing mediates this attachment? First thing, at first, leukocytes bind very weakly. First, there is weak interaction. Remember this thing, okay? And weak interaction is mediated by selectins. Selectins are the components, are the molecules which are present on the surface of leukocytes and on the surface of endothelial cells. Clear? Normally these selectins are in very, very less quantity, but when there is release of cytokines, these selectins are increased on the these are upregulated on the surface of leukocytes and vessel wall both. Okay. Important thing is, suppose this is your one endothelial cell and this is your leukocyte. On the surface of endothelial cell, you will express E-selectin. Is this clear? E for endothelium and E for E-selectin. Remember this thing. And on the surface of leukocyte, you will increase L-selectin, obviously, alpha leukocyte, alpha L-selectin. And on the surface of your endothelial cell, you will increase the ligand for L-selectin. Okay, because L-selectin is present on the leukocyte, so that you will increase ligand for L-selectin on the endothelial cell. Is this clear? And again, same, on the leukocyte, you will increase receptor or ligand for E-selectin. Now, with the help of these things, these two bind. Okay, suppose these have bind. Okay, these have binded. Now, very important. This interaction, which is mediated by selectin, is very weak. With the blood flow, as the blood flows, these leukocytes detach. First they have attached, and now they have detached. They will reattach now. Okay, this process of attaching, detaching and reattaching is known as rolling. This is very important for us because by this rolling mechanism, we bring leukocytes very near to endothelium cells. We even stick them, we even attach them on the endothelium. Is this clear? Okay. But we don't want weak interaction. Now we want tight adhesion. For tight adhesion, we have integrins. Okay. Now, let's learn about integrins. Let's move to another whiteboard. Blackboard, actually. Okay. Very important. This is your endothelial wall. And these are your leukocytes. Now, cytokines which liver was secreting will increase expression of one more thing, which is integrin. Integrins will be present on the surface of leukocytes, and the ligand for integrins, there are of two types, VCAM and ICAM. VCAM and ICAM will be present on the endothelial cells. Let me write. Okay. VChem and IChem will be present on the surface of endothelial cells. Now these two will bind. These will bind. Okay, these all will bind, and now this time the interaction is very tight. It's very, very tight. Okay, now there's no chance for the leukocyte to escape. Okay, let's read the text together. We were here. So with vasodilation, stasis, increased vascular permeability, exudation, we also have expression of E-selectins. Okay, E-selectins on endothelium and ligand for L-selectin on endothelium. Okay, because I'm talking about blood vessels, so I haven't written about L-selectins and ligands for E-selectins because they are present on the leukocytes. So on the blood vessel, you have expression of E-selectins and L-selectins. Okay, L-selectins are the ligand for L-selectins is known as sialyl-Lewis protein. Okay, and this expression is particularly mediated by tumor necrosis factor alpha and interleukin 1. I told you one more thing, I didn't write it over here, but discuss it with me. We talked about integrins as well. Integrins will be present on the surface of leukocytes, and the ligand for integrins, which are ICAM and VCAM, will be present on the surface of endothelial cells. This is clear? Okay. And E-selectin or selectins basically mediate weak interaction, and integrins mediate tight adhesion. Okay, there's one more thing that you should know. What happens to lymphatics whenever there is inflammation going in a body? You need to come to this concept again. Where did I draw it? Okay, I erased it actually. Let me draw it again. You knew that we created these spaces, and because of these spaces we were doing exudation actually. So there was so many fluid in the extracellular compartment, and you know that in this exudate we have proteins, we have leukocytes, we have offending agent as well, we have microbial products as well, and we know that lymphatics are the vessels which drain all of the excess fluid that is present in extracellular compartment, right? So all of this fluid will be drained by lymphatics, and all of the offending agent, leukocytes, proteins, debris will be going into lymphatics, and eventually lymphatics of that area will be inflamed as well. If liver is suffering inflammation, obviously there will be exudate around the liver, and this exudate will be cleared up by the lymphatics present nearby the liver, right? And when offending agent goes in these lymphatics will become inflamed. The inflammation of lymphatic vessels are known as lymphangitis. And eventually these lymphatic vessels will be draining in lymph nodes, and then these lymph nodes will be inflamed. This is known as lymphadenitis. Is this clear? So I was talking about this. What happens to lymphatics? Obviously lymphatics will be draining all of the fluid full of debris, offending agent. So eventually lymphatics will suffer lymphangitis and lymphadenitis. Okay, what happens to leukocytes? Yeah, we have actually discussed whatever happens to blood vessels. This was it for the blood vessel. Now blood vessel has very little role that we have to discuss. What happens to leukocytes? I told you we will first bring leukocytes at margins. So we will have margination. Margination is by exudation, and margination is by selectins or integrins as well. Now rolling, I told you, right? Rolling is mediated by selectins. E-selectins on endothelium, L-selectins on leukocytes, and P-selectins on platelets. Yeah, these are also present on platelets. So selectins mediate very weak interaction. So what happens when there are selectins, that there is binding of selectins and its ligand, that is sialyl-Lewis protein. There is attachment first, then there is detachment, and then there is reattachment. Okay, this is known as rolling. And why it detaches, why the leukocyte detaches easily? Because it's very weak in traction and it's affected by blood flow. Okay, now tight adhesion. Tight adhesion is by integrins. Integrins are present on leukocytes and its ligand is present on endothelium. Ligands are of two types, ICAM, VCAM. ICAM is for beta-2 integrin and VCAM is for beta-1 integrin. Just a little bit of knowledge. Okay. Next thing is transmigration. Transmigration or diaptesis. Okay, let me explain this to you again. Suppose that this is your vessel wall and leukocyte is sitting over here. Okay, leukocyte is sitting over here and it has attached to endothelium by integrins. This time the interaction is very strong. How the leukocyte is supposed to go here? How is it supposed to come here? How? This is my question. Okay, there are important kind of substances over here known as PCAM. PCAM. This PCAM mediates the attachment of leukocyte over here. Leukocyte comes and binds over here. Okay, because of this substance PCAM. Now leukocyte has to cross this basement membrane. We haven't destroyed basement membrane in creating vascular permeability, obviously. Okay, so what happens? You know that basement membranes are made up of type 4 collagen type 4. Okay, type 4 collagen. So what will leukocyte do? Leukocyte secretes collagenase. Simply, it will secrete some kind of collagenases. Collagenases will simply destroy this basement. Not all of it, obviously. Just a simple like this. Just this. Now the leukocyte is ready to escape. Leukocyte is ready to come out. This was the whole process of just bringing the leukocyte out of the vessel wall. Let's read the text together. Transmigration or diaptesis is mediated by PCAM. PCAM is platelet endothelial cell adhesion molecule. It is present in intercellular junction of endothelium. It is between an intercellular junction between two cells. This is cell 1, this is cell 2. It is present between two cells, this green thing. Okay, this mediates the migration, transmigration. Then the leukocyte pierces the basement membrane by secreting collagenase, and once it is out of the vessel wall, now how this leukocyte is supposed to go towards liver particularly. Now here comes the role of chemokines. Chemokines are concentrated in the vicinity. Okay, now these will guide the leukocyte towards the liver, okay, or the site where they are needed. Okay, so we want to spend some time on chemotaxis. What is chemotaxis? Chemotaxis is simply locomotion along the chemical gradient. This is it. Okay, now what happens? How does the chemokine work? Chemokines bind a G protein coupled receptor on the surface of leukocyte. So leukocyte has certain kind of receptor which has G protein underneath it. Chemokine molecule binds over here. It activates G protein. G protein makes second messenger. Second messenger increases the calcium influx. When calcium goes inside of cell, calcium polymerizes the actin. Now the leukocyte will become like this. Here is a lot of actin over here. and myosin is present at the back. Okay, actin is over here, and with actin over here you have created philopode, right? With philopode, this philopode will follow the chemokine concentration and it will eventually come towards liver. So this was everything about the process of bringing leukocyte out of the vessel wall towards the site of injury, towards the site of offending agent. Now in the next video we will be discussing about what leukocytes do when they reach the site of injury or site of offending agent. How do they engulf or eliminate the offending agent? I hope you have understood everything very well. Best of luck.